just a few words. So thank you so much, Professor Elinav, for being here today and for doing this talk by Zoom. Yes, we really appreciate. And so we've just heard presentations from Frank Hu from Harvard talking about multi-omics and the revolution in the nutritional epidemiology and so many work on the nurses have study and our cohorts from there. We had also a great talk from Professor Barouki about the exposome and toxicology, and just right now a talk by Professor Carlos Montero on ultra-processed foods. So I think you know also all these works, and so we are really delighted to hear you today, and so.
I'm super happy to be speaking to you today, and I apologize for not being there in person, given this nasty accident that I experienced 10 days ago and had to change all of my plans. Today I will be kind of giving you some of our insights and vignettes on the microbiome and its potential roles in precision diet. My groups and I have two groups, one at the Weizmann Institute of Science in Israel and the other at the German National Cancer Center in Heidelberg, Germany, extensively study the microbiome, which is a huge and relatively unknown microbial ecosystem that consists of trillions of bacteria, viruses, fungi, and parasites that resides within our body from the moment we are born until the moment we die. And is increasingly appreciated to impact almost any physiological function in our body. Now, they say that one picture is worth 1,000 words, and you can see here an electron microscopy picture taken at a magnification of 160,000, and in it you can see a healthy small intestine. You can see the villi, these bumpy hills representing the lining epithelia of our intestine, and above them you can see what looks like a carpet of shoelaces. This is your microbiome, and you can appreciate how vast it is and how intimately it is connected to the underlying human host cells. And we study the microbiome using an array of state-of-the-art technologies. We study the genomes of the microbes by sequencing them using next-generation sequencing by metagenomics. We study the thousands of potentially bioactive molecules that the microbes secrete through mass spec technologies, which we call metabolomics. In the last 2 years, we were able to culture up to 80% of all of these very fastidious microbes through pipelines that we and others have developed called culturomics. And we study them for causality in a variety of systems, such as organoids, organ-on-chips, and specialized mice called germ-free mice. You can see here the Weizmann Institute large germ-free facility, which are special mice that are housed in these isolators and don't carry any microbiomes of their own. They're completely sterile. And now you can transfer into them microbes or communities of microbes coming from humans and study their function in the context of human disease. And what I would like to highlight is that we can regard the microbiome, and specifically the gut microbiome, which is the most well-studied ecosystem of microbes in our body, as a signaling hub that integrates into it signals that come from the human body. For example, the human genome impacts our microbes, the human immune system impacts our microbiome, but also a variety of environmental factors and lifestyle factors, such as the ones depicted in this cartoon, send signals to the microbiome, and the microbiome integrates these very wide array of signals and communicates them to the host. And we believe, and we spend our lives trying to tease apart the alphabet of the language by which we and our microbes communicate with each other in health. And we try to understand what goes wrong when these communication networks go altered, that leads to susceptibility to important and common multifactorial human diseases. And given today's topic, I chose to give you a few vignettes of maybe our major focus in the last decade and a half of microbiome work, which relates to how diet impacts the microbiome and through the microbiome impacts metabolic health. And I think this crowd needs no appreciation of the obesity and the associated metabolic syndrome epidemic that we are facing as a species. In just 150 years of post-industrial revolution, and such a short time span does not imply that genetics, which do impact the metabolic syndrome, are the major driver, but rather environmental and nutritional factors. And and you've heard other talks mentioning and discussing several human changes that accompany or associate with this huge surge in metabolically related disease, such as the huge increase in consumption of sugar in both the developed and less developed world. And I'll mention briefly also some of the non-nutritive replacements which we've come up with to sugar that have been exponentially consumed and may have independent effects on this epidemic. And in my talk, I would like to highlight three insights, three eureka moments that we came up with regard to our understanding of how diet impacts the microbiome and downstream metabolic health or metabolic disease. Now, the obvious avenue of impact is diet impacting, or the composition of diet, or the components of what we eat impacting the microbiome. And of the many environmental factors that shape the microbiome, diet by far is the most dominant. And through these dietary compositional interactions and effects, the microbiome can change and affect the downstream host and host metabolism. But what we've discovered in the last decade was that also dietary timing has independent and fascinating impacts on how the microbiome composition and function are shaped, and these have independent effects on metabolic health and metabolic disease. And the third avenue of dietary impacts on the microbiome that I'm going to briefly mention today relates to long-term dietary patterns, which we were surprised to find have their own impacts on the microbiome and their own impacts on our ability to maintain healthy or unhealthy metabolism. And I'm going to give you really one vignette of each of these avenues which we explore till this day. And I'll start with dietary composition. And when I started thinking about how our dietary preference, our daily dietary preference, kind of impact our health, or how we hope they impact our health, I started reading many of the old papers that form the basis of everything that you and I do in our daily basis when we try to match our diet with an hope of becoming healthier. And what I realized was that everything that we do is based on a few grading or scoring system which give different numbers to different foods. For example, calories are such a grading system which give different values to different food components, and all of us intuitively or non-intuitively try to mix and match these caloric values to come up with what we hope would be a healthy diet. An even more widely used grading system of food that is at the basis of almost anything that we do nutritionally and is not strange to this crowd is called the glycemic index. And the glycemic index is based on very small human trials conducted in the 1970s, which usually included 10 healthy individuals who were given an identical food, and then their blood sugar levels were measured for 2 hours after they consumed the food. For example, if you were to take 10 healthy individuals in this crowd and you would kindly give them an identical piece of celery, the blood sugar level spike would look like something in the black graph. It would be given a value, and this would be the glycemic index of celery. Now, if you were to take the same 10 healthy individuals and now kindly ask them to consume an identical piece of chocolate cake, their average spike in blood sugar level would be higher, just like the blue graph. It would be denoted a higher number, and this would be given as the glycemic index of chocolate cake. And if you were to look in your smartphone, you would find endless tables that provide glycemic index values to any food or food component on Earth. And take my word for it, if you need, any diet or almost any diet that you've ever tried, whether you went to a dietitian, a physician, or bought a book at the airport, is based in one way or another on this glycemic index value or system. Now here comes the big problem. When we repeated these small-scale studies from the 1970s using much larger numbers of individuals and much more modern technologies, we found that these notions were inherently incorrect. So for example, the first clue to a problem in our way of thinking came in a study we published almost a decade ago, focusing on some non-nutritive sweeteners. This was a mouse and a human-based study. And for example, when we took a group of healthy human volunteers and we gave them an identical amount of saccharin and measured their glucose spiking, their glucose elevation post this consumption, to our amazement, we found that half the people spiked on saccharin consumption, while the other half didn't care, meaning that the average spike did not represent what we saw at the level of the individual. And this led to a much larger and well-controlled human clinical randomized trial looking at all 4 commonly consumed sweeteners, which was published just last year, and we saw exactly the same thing. People's responses to these artificial sweeteners were highly personalized and dependent on the microbiome, and this led to a rain of follow-up papers, which culminated just less than a month ago in the WHO recommending for the first time against the widespread use of artificial sweeteners for metabolic indications, following all of this emerging data that these compounds are not inert to the human body. But our conclusions spanned much more than just this one early example of food supplements. When we looked at 1,000 healthy Israeli volunteers and gave them one of several test foods, for example, 50 grams of glucose or an identical piece of white bread in the left graph, we saw that the average glycemic postprandial spike to each of these identical test foods of the entire population was exactly the glycemic index of that food. So inherently, the glycemic index seemed to be correct as an average indicator of response. But when we looked at the individualized response to this identical food, we were amazed to find that different people respond very differently when they eat the same exact food, which already told us intuitively that the notion that we can come up with a healthy diet based on the characterization or the quantification of food may be inherently flawed, and maybe we need to replace it with actual measurement and quantification of the people that eat this food. And this was not just true for the test foods that we've tested in these 1,000 individuals, it was true for thousands of real-life food, which we've measured in the right panel, and in all of them, the variability in people's responses to the same exact food was unimaginably high. And we've done this in over 100,000 people up to now. So this seems to be a very robust observation. And this eureka moment gave rise to a very ambitious project, which we termed the Personalized Nutrition Project, in which we took these 1,000 people and kindly asked them to give us a week of their life, in which we've sampled them and collected, at the time, an unprecedented amount of personalized data, including gut microbiome sequencing, a very deep one, which is what we do in life, blood tests, many questionnaires, anthropometrics, a continuous glucose monitor that measured their blood sugar levels every 5 minutes for an entire week. So just to give you a flavor, in these small-scale 70s experiments, 10 individuals were skin pricked for six time points. Here you have 2,500 measurements per person being done in 1,000 people, giving you a very large data set. And we've included a smartphone app that gave us a very nice point of care data on whatever these people were doing in their daily lives. And all of this data was then collected by our students and used in machine learning and deep learning AI algorithms to train them to predict a person's glycemic response to any given food, including foods that they've never consumed in the week of follow-up. And to summarize 7 years of work into one slide, on the left panel, you can see the predictability of what we do today. When we want to lower our blood sugar levels, we go to a dietitian or to a clinician, and we ask for advice, and we're usually recommended to reduce the amount of carbohydrates in our diet, which has a positive predictive value, but it's very low, it's 0.38. But when we use this data-driven, science-driven, individualized approach, we dramatically increase our ability to predict a person's glycemic response to individualized food. You can see in the middle. And as you have to do in these cases, at some point we froze the machine learning algorithm and we applied it on a fresh cohort of individuals who were not part of the training, and we reached similarly high results, which made us very happy. Now, at this time, it was time to put our computational insights to the real-life test. So we've gathered a group of pre-diabetic individuals. We put them through our system, and we this time asked the computer to prescribe an individualized good or bad diet for each of these individuals. Of course, the diets were different between individuals because they were personalized and very counterintuitive. And then we asked the individuals to eat their individualized good diet for a week, then their individualized bad diet for a week while we followed them. And to our surprise, even a week of seemingly counterintuitive intervention, which could include some chocolate in some cases or sushi being bad in others, resulted in a marked improvement in people's glycemic results. And when we visited the algorithms in retrospect, these are unbiased algorithms that don't ask for our permission on which feature to use, we found that some of the features were coming from the microbiome. And in fact, today we've crystallized the system to only use the microbiome in a limited set of clinical features to get at comparable predictability. And these results, which were quite a change from the known dietary paradigm, were repeated by multiple cohorts around the world in different ethnicities, which generally generated the same insights. But it was time to put our system to the long-term test. This is the million-dollar question. So we've recently completed a randomized clinical trial in which we've enrolled 200 pre-diabetic individuals. We randomized them either to the best of the best intervention, Mediterranean diet, low carbohydrate, the best recommendation we can give as physicians, and the other half to this counterintuitive, data-driven, personalized algorithm diet. We've extensively followed them for a year, including half a year of continuous glucose measurement. And you can see on the bottom an example of 3 of the major glycemic readouts, in which you can see in red the control Mediterranean diet group with the mild but significant improvement in glycemic control, which has been well described in the last 2 decades. But you can also see in green the personalized diet, which outperformed the state-of-the-art diet by quite a nice margin, with the results maintained, given the fact that the diet is much more flexible and easy to follow over a long period of time as compared to the harsh restrictive diets that we currently recommend. And just to mention that this doesn't stop with diet, even in other contexts, for example, over-the-counter probiotic consumption, which we've assessed in humans in the most invasive microbiome study to date, I think, we found that colonization of probiotics was also highly individuals, with half the people consuming these probiotics in which the microbiome, the indigenous microbiome, resisting their colonization, even short-term intermittent colonization, and the probiotics ending within a few minutes in the other side, while in the other half, the microbiome was more accommodating. And in this group of people, we could see that the probiotics could induce impacts on the host. And we don't stop there. We are now following up on several clinical trials, including one trying to assess childhood obesity with Italian collaborators, and a very ambitious trial in which we try to adapt the same insights to see whether we can harness diet in a data-driven measurement approach to tackle non-metabolic consequences, such as inflammatory readouts in Crohn's disease. So this is just an example of how dietary content impacts the microbiome and how we can utilize it to hopefully affect the metabolism. But as I've mentioned, we've also identified that dietary timing has an independent effect on the microbiome. And in this study, which also started over a decade ago, we were serendipitously able to identify that the gut microbiome features a very strong diurnal activity. In other words, it changes its composition and function over a 24-hour cycle. You can see here an example of the composition of the microbiome. This is the large intestinal microbiome, in which multiple species of microbes feature a very strong and statistically significant diurnal or circadian activity. You can see on the right an example of one of the commensals going up during the night, down during the day, up during the night, down during the day, forever, basically. But this was also diurnally shown for the function of microbes. When we looked at microbiome function, you can see that multiple functions, you can see in the heat map, multiple functions go up during the day, down during the night, and so on and so forth forever. And even complex microbial activity, such as flagellar activity, which bring together different metabolic pathways, feature a very well-coordinated diurnal activity, going up or down at identical times during a 24-hour cycle. Now, this was very surprising for us, because if you think about it, these gut microbes live completely in the dark. So how do they know that it's day and night and change their composition and behavior so reproducibly? And this led to a multi-year effort. And at the end, we found out that one of the major influencers on this diurnally shifting microbiome activity in the gut is the timing of our diet. So for example, if you take two groups of mice, and in one group you force feed them only during the nighttime, this is the left upper panel, and in the other group you only feed them during the daytime in the right panel, and you measure over a 24-hour period the composition and the behavior of the microbiome, you can see that you can shift these behaviors by 12 hours just by shifting the timing of the diet. Now, why is this important? And we've discovered this, the first hint that this diurnal microbiome behavior is important to host metabolism by performing a quite unique set of experiments, both in mice in the left and in humans in the right, in which we've subjected them to a very severe disturbance in their circadian activity. In mice, we've created a severe state of jet lag through known mechanisms. In humans, we've taken a group of students and we flew them around the world while asking them to collect their poop before, during, and after they were jet lagged. We've sequenced the microbiomes and we found that a marked disturbance in the diurnal activity was associated with a microbiome dysbiosis, with a disturbance of the microbiome. But to reach causality, we then transferred the jet-lag microbiomes or the non-jet-lag microbiomes from mice or from humans into germ-free mice, the same mice that I've mentioned before, to not have any microbiome of their own. And to our amazement, these germ-free mice developed a tendency to develop more diabetes and more obesity as compared to mice that were transferred with a non-jet-lag microbiome, which tells us that maybe some of the very well-known susceptibility of shift workers that's been known for decades in human medicine to develop diseases such as obesity, diabetes, and even cancer is driven, at least partially, by these microbial disturbances. Now, we didn't stop there, and we've continued to follow up a project in which we looked at other regions of the gut, for example, the small intestine, and we've similarly shown that the small intestine microbiome featured a very strong diurnal activity, different microbes, different behaviors. Nonetheless, they change across a 24-hour period in a very reproducible manner. Now, why was this important? In this case, it was important because the small intestinal microbiome had a very great impact on the small intestinal immunity. For example, the antigen presentation performed by intestinal epithelial cells was very diurnal and very much dependent on that crosstalk, on the tango between the epithelial cells in the gut and the circadian microbiome, because if you disrupt the microbiome by giving antibiotics or by performing these experiments in germ-free mice, you completely abolish the normal immunity induced by these epithelial cells in the small intestine, and this could be shifted by the similar approach to what I've shown you before of controlling the timing of diet. So by the timing of diet, amazingly, we could control gut immunity through these circadian axis of impacts. And why is this important to our health? We discovered that this circadian disturbance in epithelial cell immunity leads to a cascade of disturbances in other downstream immune circadian activities, leading to a loss of tolerance and an impaired barrier function in the small intestines of these dietary altered or jet-lagged mice, which subjected these mice to a great susceptibility to immune diseases such as Crohn's disease in the small intestine. So this is just an example how dietary timing, through surprising, I think, mechanisms, could impact our microbiome in ways which would affect our metabolism, but also other immune consequences to the mammalian body. And my final example would relate to long-term dietary patterns, which we also accidentally stumbled into and found to have impacts on the microbiome and metabolic health. And specifically, we decided to focus in this project on a very well-known form of obesity, for sure, to this crowd, which is called relapsing or yo-yo obesity, which actually is the obesity shown in the majority of people suffering from obesity worldwide. And this is the context in which a person develops obesity for whatever reason, goes on one of thousands of diets that are all involving caloric restriction and helpful in the short term, loses weight back to their baseline level, but within 12 months, 80% of these people rebound back to obesity, and from cycle to cycle, they gradually develop more and more obesity until they become formally obese. Now, if we know increasingly the pathogenesis and the factors impacting the pathogenesis of obesity, we know much less about the factors regulating relapsing or yo-yo obesity. So we decided to explore this in mice and to see whether the microbiome has anything to do with it. And we've developed 3 different mouse models recapitulating human relapsing obesity. For the sake of time, I'm just going to show you one of these models. They all ended up showing the same. In this model, we use multiple groups of mice. You can see on the top, the blue group are mice that were fed with a high-fat, high-sucrose diet and developed continuous obesity. The black group were mice fed normal chow and not developing the obesity. These were kind of the extreme groups. And then take a look at the red group. These are mice that were fed a high-fat diet, gained weight just like the blue group, but at some point we switched them back to normal chow. They dieted back to the black group. Then we exposed them again to another cycle of obesity, and then a third cycle of obesity. Now, if you focus on this third cycle of obesity, you can notice that mice that were developing obesity for the third time, although they started from the same exact weight and getting the same exact chow, were becoming more obese as compared to the green mice that were in their second cycle, and as compared to the orange mice that were in their first cycle, meaning that we can corroborate the exaggerated weight regain that characterizes human relapsing obesity. And this was not only true for weight, there were multiple metabolic parameters which were gradually deteriorating from cycle to cycle, as you can see in the bottom of the panel. Now, the biggest question was, what in the mouse remembers that mouse was once obese and predisposing it to an exaggerated weight regain phenotype once it was re-exposed to the obesogenic conditions. So to explore this, we decided to focus on the point which we call the point of nadir, in which mice that were cycling went back to their original healthy weight. So at this point, the weight was not different, but something in the mouse was remembering the previous weight event, and we measured many different inflammatory, metabolic, and hormonal features at this nadir point, and we found that all of them completely normalized following a successful diet. All of them, but one feature, the microbiome. The microbiome, the gut microbiome in these successfully dieting mice remained in an intermediate configuration that was somewhere in between the obese disturbed phenotype and the lean non-disturbed phenotype. You can see that the red configuration in different readouts was between the blue and the black configurations, meaning that somehow it was not reverting back to its healthy configuration following a healthy diet. Now, this is a nice observation, but it doesn't prove causality. It doesn't prove that the microbiome is driving this weight exaggerated effect. So since we pride ourselves on trying to contribute to the field ways to examine causality, we performed a number of approaches to prove this. I'm just showing you one for the sake of time. We took, at the nadir point, microbiomes from previously obese and successfully dieted mice, or mice that were never obese before, and we transferred them into germ-free mice. And to our amazement, mice that were transferred with this microbiome coming from successfully dieting mice developed more obesity and more diabetes when subjected to metabolic disturbances as compared to mice that were transferred with microbiomes coming from never obese mice, suggesting that something in the microbiome was remembering the past obesogenic effects and was driving the exaggerated post weight regain phenotype. Now, of course, the billion-dollar question was, what is driving this memory in the mouse? This led to a very deep dive into the metabolomics, into the small molecule repertoire secreted by the different microbiomes in successfully dieting obese or never obese mice. And to make a very long story short, we got very lucky in this case and found that in our facilities, there was one pathway, one function of the microbiome that was significantly suppressed in post-dieting mice. And this was accompanied by a marked reduction in 2 products of the pathway, 2 isoflavonoids that were suppressed in these post-dieting mice. And this brought the possibility that maybe the lack of these 2 isoflavonoids was actually driving the bad metabolic memory that was predisposing these mice to an exaggerated weight regain in the next time they're exposed to an obesogenic condition. Now, to test causality, we now replenish these 2 isoflavonoids that were missing in these post-dieting mice and subjected them to another cycle of obesity. And indeed, we could completely avoid and prevent the emergence of an exaggerated weight regain phenotype, which proved that, at least in the mouse setting at out facility, these isoflavonoids and their deficiency was driving the effect. And we went even further to try and understand what they're doing. And the answer was that these isoflavonoids were swimming into the mouse where they were signaling into adipose tissue cells and preventing them from performing the normal decoupling of ox-phos mediated by the enzyme UCP1, and therefore these adipose tissues were generating less heat and making more fat as compared to mice that were not having these conditions. In other words, these two molecules were helping mice generating more heat and less fat, and when they were absent, they were driving the exaggerated weight regain by disturbing this decoupling event. Now, interestingly, when we published this study, just a month after came this New England Journal of Medicine study on relapsing obesity in humans. This was a long-term, decade-long observational trial, a very important one, not related to the microbiome in any form or shape, but it taught us that that relapsing obesity in humans is not only a very common feature, but an independent risk factor for the emergence of diabetes and for coronary events, and even for all-cause mortality and cardiac mortality. And this promoted us to generate a human study called the Yo-Yo trial, which is ongoing as we speak, in which we try to adapt all the technologies and insights that we've developed in mice to study whether the microbiome also participates in human yo-yo obesity, which at least theoretically could represent attractive preventive and therapeutic opportunity for us. And just to wrap up this talk, I hope I've given you some sense of how we approach the study of host microbiome interactions in metabolism, and the promise of mechanistic and causative insights in progressing beyond the currently utilized one-size-fits-all microbiome treatment approaches, which are kind of limited, to say the least, in their evidence base, including prebiotics, probiotics, and fecal microbiome transplantation, especially in the metabolic context, with more data-driven, personalized, and science-driven microbiome utilizing approaches. I've given you a couple of examples that may enable to reach better results to the individual, and I am hopeful that within the next decade, these new modalities would be gradually integrated into the precision medicine clinical shelf. And I will finish this talk by thanking the many people from all over the world who have joined me in this journey and have been an important part of these really ambitious and difficult trials at the Weizmann Institute of Science. The many collaborators, the too many collaborators to mention, and patients and participants that are making all of this work possible, and of course the funding agencies that are making all of these efforts possible. And just to finish, I'll mention that in the last 4 years, I've also established and had a unique microbiome and cancer division at the DKFZ, hosting amazingly talented students and postdocs. And there we try to adapt the technologies that we've developed at the Weizmann towards the study of possible impacts of the microbiome on the development, progression, and response to treatment of different cancers. And with that, I think I'll stop here. Thank you.